Yeah, nice to meet everybody. New postdoc PBRC working in the Amen lab, and I'm going to tell you a little bit about today some of my research from my PhD work, and then how that is leading into some of the projects that I'm doing here in the Amen lab. And I'll preface this by saying, please feel free to interrupt throughout the talk if you have any questions, and we can just address them right there, because I'm going to start a little bit more ecological information heavy. But kind of starting off here, I really like this graphic over here on the left, which is from Nelson et al. 2023. Craig Nelson actually works here at UH. And ultimately, what I'm going to be discussing today are kind of the interactions within the microbial parts of the ecosystems that are driving a lot of these fundamental ecosystem functions. And so on coral reefs specifically, we know that microbes are maintaining high biodiversity by forming symbioses with different organisms. They're recycling a lot of our dissolved primary production, so they're making sure that that's available for higher trophic levels. They're enhancing nutrient availability through remineralization, and then on coral reefs specifically, they're actually facilitating net accretion, which is the growth of the reef. And so this talk is going to kind of focus on how these different microbial interactions impact these community assembly and resilience processes, right? And in the first bit, we'll be talking about some of my PhD work, which is how viruses and microbes build and dismantle coral reefs. And then next, I'll talk about very briefly some of the projects that we're working on here at UH, which involve how bacteria modulate energy transfer up trophic levels and how fungi and bacteria interact to contribute to ocean carbon cycling through degradation of marine snow. So let's jump into coral reefs, first of all. If we can put a value on an ecosystem, then we can say that coral reefs are reasonably generating about $400 billion a year through their different ecosystem services. They're providing resources directly and indirectly for about 1 billion people, and they're hosting about 25% of all ocean life. So I don't really think I have to convince you that coral reefs are important. They're also degrading through a variety of global pressures. We have our ocean warming, acidification, things that sound pretty familiar to you, as well as all of these local pressures, which are more things like overfishing and pollution and coastal development. And one of the things I'm going to try and convince you of in this talk is that most of these actual impacts, while they're generated by anthropogenic sources, they're actually mediated at the fine scale by microbes. So we're going to talk a little bit about how microbes contribute to building and degrading coral reefs. And I'll start here by saying that coral reefs are really, really hard to build. These are extremely complex systems. See if I can play this video here. You can see in this video that there's quite a bit of coral cover, lots of structure, some pretty happy fish, very, very clear water, indicating a lot of these nutrients are being recycled. And to maintain this really complex system, you really need a positive feedback loop to be able to be driving this in a certain direction. And that positive feedback loop starts at the base of the food web, where we have this really awesome tight partnership. One of the reasons why corals are better than mosquitoes is because we have this really tight partnership at the base of the food web between corals and their algae endosymbionts. And what's really happening here is that these algae are producing a lot of photosynthetic sugars, and then all of that energy is going into these corals, which they use for energy in an ecosystem building process that then builds up this really awesome structure. Right. And so once again, I can't stress this enough that this partnership, as we'll discuss later, the fact that oxygen and sugars are being recycled within this partnership and used to build an ecosystem process is the really, really awesome part of coral reefs. So coral reefs are then producing all this structural habitat, which provides a home for all these animals, including many that eat algae, which is one of the main competitors with corals on the seafloor. And lastly, if we look at the unseen component here, we have our viruses in pink, we have our bacteria in blue, and what we see on a healthy reef is a relatively high abundance of these free viruses and a relatively low abundance of our free microbes. And we call this this metric the virus to microbe ratio, and we use this as an indicator for how healthy a microbial community is on reefs. And I'll use it throughout the talk, so I want you to remember that. But basically, all you need to know now is that there's a lot of viruses relative to fewer bacteria on a healthy reef. And this is really indicating that viral predation is controlling those microbes and keeping our microbial abundances from growing out of control. So let's continue on that microbe train. We know that microbes consume organic matter, and that this can have really foundational impacts on our ecosystem conditions, right? So organic matter comes in many forms, comes from natural sources, like whenever something dies, the exudates from algae, the spawn slicks from corals, and then there's also anthropogenic sources of organic matter. We have mariculture effluent, we have sewage pollution, agricultural and riverine runoff. What we know in all cases that whatever organic matter you have, when you add it to a coral reef or to any other really ecosystem, microbes do what they do best, which is to eat. And the process of eating, which is called respiration, ends up taking in oxygen and outputting carbon dioxide. Now this in the oceans draws down O2, and it actually draws down pH as well. So as you add organic matter, my point here is that these microbes, microbial consumption of this organic matter can actually can change the face of the ecosystem and the conditions of the ecosystem through deoxygenation and acidification. And the important thing to note here is that there is a large set of microbes that like these conditions, but there's not a large set of macroorganisms that like these conditions, right? So organic matter is one of our kind of big beasts that we're dealing with here, which segues us into how a coral reef degrades, right? Our lab has studied this positive feedback loop of coral reef degradation that we call DDAM for a couple decades now. DDAM stands for dissolved organic carbon, disease, algae, and microbes, which we'll go through right now. And really where this starts is when human impacts like nutrient inputs and eutrophication and overfishing, such as the removal of herbivores, really kind of give a competitive advantage to algae on the seafloor over corals. And algae don't have this tight partnership with corals, where they are using this ecosystem energy, this productivity, and funneling it into an ecosystem building process. Instead, they release a lot of their photosynthetic production as labile or bioavailable dissolved organic carbon into the seawater. And this is probably about up to 50% of their production goes into the seawater, where it then feeds these microbes. And microbes love organic matter and they eat. And we see shifts in these communities that become more cobiotrophic and more pathogenic. We also see this interesting shift in the viral communities on these degraded reefs, where suddenly this virus to microbe ratio, or the abundance of viruses relative to microbes in the ecosystem goes down. And we'll talk about why this is, but ultimately what we have discovered is happening here is that viruses are switching from a canonical lytic predatory strategy, where they explode the microbes, to one where they just are piggybacking off of the microbial success here. As these microbes are growing really, really fast, the viruses are like, why don't we just integrate into your genomes and tag along? This increased incidence of pathogens are then causing disease among coral reef macroorganisms, which kills corals and frees up more space on the benthos to drive this positive feedback loop forward. There's one more piece of this. I did mention that these algae at the base of the food web do not have a mechanism for actually building ecosystems with, they're just releasing a lot of their DOC into the water. The same goes actually for their oxygen. They're releasing oxygen at a much higher rate than corals, and a lot of this oxygen actually supersaturates on the surfaces of their tissues and then bubbles off to the atmosphere. So algae can lose about 25 to 28% of their oxygen production to the environment because of this. And what ends up happening is the combination of this increased microbial respiration and this loss of oxygen through algae bubbling leads to hypoxia that continues to kill reef macroorganisms. So this is a pretty fierce cycle, right? And we call this whole process microbialization, which is this kind of slow transition from a macro-dominated ecosystem to a micro-dominated ecosystem. And one of our main questions and one of the main focuses of my PhD was trying to figure out how we break this feedback loop and try and enhance reef function. So we know quite a bit about how this works at the molecular level as well. On the left here, you're seeing a representation of a healthy coral reef ecosystem, which is represented by corals down here. And what we know is that, and which makes sense, is that microbes, when they have more scarce resources, so less resources at their disposal, are going to have to be more efficient, right? And so in this case, these microbes have low extracellular food sources, and so they're using metabolic strategies like the Emden-Meyerhoff-Parnas pathway that generate a lot of ATP, but it happens slower. So they're trying to eke out as much as they can from these carbon sources. And they end up fully oxidizing these carbohydrates to CO2. And then that results in kind of these lean microbes. We can think of them as smaller, leaner microbes with lower biomass. And these are really good prey for viruses. And so we see a lot of lytic predation happening here. On an algae-dominated reef, we see a switch. So suddenly we have a ton of surplus carbon on the outside. And so we see these shifts towards microbes that are using different metabolic strategies. Suddenly they don't have to be as efficient with their resources. And so instead, they can use these alternative metabolic pathways that just kind of burn through that carbon. They generate less ATP, but they do it faster, right? But because they're generating less ATP, they can't actually always get all the way through the TCA cycle. And a lot of their metabolic intermediates end up getting shunted out into biomass. So these microbes actually are physically getting larger because of this phenomenon. So we see microbial biomass on these reefs increase. We see these shifts in their metabolic potential. And then the last thing is because they're growing so fast, growth rates are really high, we do see a shift in these viral communities as well, where they tend to favor lysogeny and integration to kind of piggyback along microbial success rather than lysis. So ultimately, I've given you a lot of information here. We think this is really coming down to these two levers here, our electron acceptors, which is our oxygen, and our electron donors, which are our sugars. So we think pushing the system in either of these different ways really impacts how microbes are acting in an ecosystem. And to kind of summarize a little bit further, before we get into the actual data, we have these kind of two different trajectories that a reef can go, right? On the one hand, which we call viralized for no good reason other than the fact that we like viruses, the energy from the sun goes into the corals and then gets funneled up this really rigid trophic web. And so our energy is being stored in these kind of tight compartments, and we have this really tight cycling. Microbes are kind of like in control under this viral predation. And then we move over to this other microbialized side of things. And we have the sun's energy going into algae, which is then being released into the water and feeds microbes, right? So we have this kind of more loose cycling where this ecosystem progressively becomes more and more dominated by microbial states. Okay, summarizing all that, I gave you a lot of information, but we're going to talk about the fun stuff now. Healthy reefs have a lot of primary predatory fish and viruses, and coral reefs are retaining oxygen and building structure. By adding a lot of organic matter and removing the fish, we release the predatory controls on algae and microbes. We drive this microbialization feedback loop. And then this is reinforced by changes in microbial metabolisms, ecological interactions with viruses, and the changes that we see to reef biogeochemistry. So the working hypotheses I kind of went on for my PhD revolved around oxygen and DOC. We kind of thought that the ratio of electron donors, so our DOC, dissolved organic carbon, to electron acceptors, which is our oxygen. We call this EDAR for no good reason other than it was easier to say than the ratio of electron donors to electron acceptors. We think this determines reef community structure, and that when EDAR is high, we expect it to be more microbialized, and when EDAR is low, we expect these systems to be healthier. And finally, kind of our mechanistic part of this is we expected that by moving a reef community up into the midwater, where you have more oxygen and you have lower DOC, we expected we'd be able to reduce EDAR and therefore microbialization. So the way that we decided to do this was these tools called coral reef arcs, which basically get off of this benthos where you have a lot of these metabolic processes happening up into more stable midwater conditions. And so we had to figure out how to build these midwater reef systems. First step is figuring out how to collect and move reef biodiversity. We know that there are many organisms, not just corals, that contribute to reef function. A lot of these are cryptics, they live inside the reef, and these guys are recycling nutrients. They are providing shelter for these zooplankton, which is food, and they are helping export organic matter and basically process organic matter. And some of you might be familiar with this project called the ARMS project. This was an NSF PIRE project called Autonomous Reef Monitoring Structures, where we basically built these standardized plates or settlement structures and then deployed them all over the earth, thousands of them, for a few years, and then sampled them. And the goal was to try and census coral reef biodiversity. And what they found here is that they could collect about 80% of coral reef biodiversity, which is pretty good. So that kind of demonstrates that we have this tool to collect and move a lot of this cryptic biota. We started making these specifically out of limestone because it kind of gives us the ability to change the internal characteristics, but it does pretty clearly show that these things get fouled with cryptic biodiversity and gives us a tool to move this stuff. Next was we had to design this midwater reef system. These are the criteria we used over here. Nobody's ever done this, so we didn't really kind of know what we were doing first, but we wanted it to have this internal reef environment that would kind of fill the role of the inside of a reef. We needed it to be able to move up and down in the water so that it could leverage these improved conditions in the midwater. And then, of course, it needed to be logistically feasible, deployable by boat and divers, and robust to ocean forces, which they were not always, but they tried to be. We came up with like a bunch of different designs. I think we built 10 or something like that, but we ended up landing on these two that we deployed throughout the Caribbean and then Madagascar and Saudi Arabia. And I'm going to show you one of those field experiments today. But basically, these are the different systems that we use for different applications, depending on the site, etc., and some pictures of us actually getting these things out there. And so for our first big field experiment, we're working here in Puerto Rico off the coast of Isla Vieques, which is a little island off of Puerto Rico's east coast. And we deployed two arcs, and this is in a little bit deeper of water. It's about 60 feet deep. And then the arcs are floating, right? So we had them floating at about 10 meters. The tops of the arcs were about 10 meters, so 30 feet. And we set up these seafloor control site plots also at 10 feet, so at the same depth, but closer to shore so that we could actually get those depths right. We moved 100 corals to each arc and to each control site. And then we kind of had the hypothesis that better microbial and water quality conditions on the arcs would lead to better survivorship of corals. And we measured a ton of stuff. We sampled over 2 years. Some things we measured were flow, light, dissolved temp or dissolved oxygen and temperature, as well as all the macro stuff, which I'll talk about as well, corals, fish, etc. And then, of course, our microbial variables, so these viral and microbial abundances, microbial biomass, inorganic nutrients and DOC, and total DNA. And I'm only gonna have time to show you some of these things today, but let's talk about what happened. Does anybody have any questions before I go into data? Yeah.
Sort of, how big are the arcs?
So they're about 10 feet in diameter, and they're like 750 pounds, and they're a huge pain in the ass. But yeah, there's quite a bit of surface area in there and on the tops as well. And we put the arms in there as well. So back to our microbial stuff. It's a little hard to see this up here, but I want to remind you before I go into this data of what we expect to see microbial communities look like on a healthy versus a degraded reef, right? Especially from the perspective of abundances, we expect healthy reefs to have this high VMR, which means that there's a lot of viruses relative to microbes. That's indicating viral predation. On the degraded reefs, we expect to see a lot of microbes relative to viruses, so a low VMR, which is indicating that these viruses have shifted their strategies towards a lysogenic or a temperate strategy. And then finally, we expect to see smaller cells in a healthy environment because this means that less of the energy is being shunted into that microbial compartment and more is available for macrobes. So digging in, you're looking here at a box and whisker plot with virus to microbe ratio on the y-axis. This is data shown over all time. And then this plot right below it is the same data, but just showed at each time point. It's broken up by arc and control. And as you can see, the virus to microbe ratio is significantly higher on the arcs than at the control sites. And it was pretty consistent like this throughout the experiment. When we look at what makes up the virus to microbe ratio, so these abundances of our viruses and of our microbes, we can see that virus-like particle abundance was significantly higher at the arcs, and significantly microbial biomass or microbial cells were significantly lower at the arcs.
How did you do the quantification of the viruses versus bacteria?
At fluorescence microscopy. We tried flow cytometry as well. Doesn't pick up. I mean, viruses are just kind of a lot of those smaller viruses are in the various side of the noise side of the FCM. So we did epifluorescence microscopy here
With a double-stranded DNA stain.
with cyber.
And where did you sample? Did you sample within the arc? Was it how far above the coral? Was it inside that structure, outside the structure?
Yeah, so we developed a sampling regime basically that was targeting basically in the area that the corals were living in, so near the top of it. And what we did is we took, a couple of technical replicates on one side, and then another replicate on the other side, and then we did north, south, east, west, so cardinal directions. So my whole point there is we did end up trying to, assess whether there were differences across the arcs, and there weren't significant differences whether or not we collected it from slightly inside the arc, slightly outside the arc, and at the cardinal directions. And finally, our last plot is looking at mean microbial cell volume. We quantified this with DAPI. It wasn't with Cyber because Cyber is too big of a halo. But the mean microbial cell volume on the arcs was significantly lower than it was at the control sites, which is kind of confirming in our minds that these sites are less microbialized than the control sites. These are the metrics we're using. I also have a bunch of DNA work that I'm not going to be able to show you, but we can talk about that later. So what are one of the reasons why these things were so successful? I mean, one of them is definitely the water quality conditions. The midwater arcs were definitely displaying better water quality than the control sites were. We can see that here in the left plot. The arcs had faster average flow speeds. When it comes to daytime light intensity, the arcs had higher average light availability. And this is really because of turbidity in the water column at the control sites. And arcs also had higher average dissolved oxygen. And we know that all of these things are associated with improved physiological success and performance of different macroorganisms. All right, let's take a look at EDAR, which is not going to be a very satisfying answer, but basically what we did, what you're looking at here is dissolved organic carbon at the arcs and the control sites over time and over all time. And while we can see that there were differences in DOC, specifically it was lower at the arcs, we don't see this, we don't see these significant differences over all time. And this is probably for methodological reasons. We're measuring bulk DOC here, and microbes are actually feasting on more of the labile component. We don't have a great way of measuring labile components of DOC yet. And so what we're probably seeing here is an artifact of just looking at this whole dissolved organic carbon community rather than the interesting stuff. But oxygen did have some more cool findings here. This is a density plot, so you're looking at basically the frequency of points here, and then this is our dissolved oxygen axis here. Arcs are here, control sites are up here, and then we also have our dark colors are nighttime oxygen, and our light colors are daytime oxygen. The cool thing here is that you can see that with the arcs, you get this really strong peak during the day and at night with dissolved oxygen, so it's staying pretty high all the time. Whereas you start to see this day-night shift in dissolved oxygen at the control sites. And this makes sense. We see this all the time on degraded reefs, where you have higher microbial biomass, which draws down oxygen at night because of increased respiratory demand. And you have higher daytime oxygen, which is because of all this algae photosynthesis. This, we did not see verging into hypoxia here, but it does very frequently get to hypoxic conditions on degraded reefs, which can really challenge a lot of different macroorganisms. We looked at the relationships between all these different parameters. This is a Spearman's correlation matrix where the green bubbles are positive associations, brown bubbles are negative associations, and the asterisks are significance. And I want to pull out a couple of these interesting ones that we found, one of which was that there is this really strong positive relationship between the virus-microbe ratio and dissolved oxygen, which is really kind of hinting to us that this metabolic switching of our lysis to lysogeny, so our viral infection strategies, is somehow related to oxygen, which I actually think is, we have some ideas for why that might be the case molecularly, and I'd be happy to chat about that if anybody's interested in viruses, but I will not bore you if not. Then the other thing is kind of confirming this microbialization dynamic. As we see dissolved organic carbon go up, we see the number of viruses go down, which is really indicating to us that as this organic matter in our system increases, viruses are switching these metabolic strategies towards lysogeny. And finally, last part before I show you some photos, I know you're like, show us the photos. We ran a random forest, and this really showed us to try and determine which were the most important predictors of whether or not we were looking at an arc or a control site. And the most important variables here were the dissolved oxygen, the VMR, and the flow. Okay, let's look at some videos. This is in the beginning of the experiment when we first deployed the arcs. This is 18 months later. You can see that there's a pretty remarkable transformation that's occurred. We have a lot of diversity, we have a lot of fish, we have a lot of activity going on in these structures. And so this is kind of, you know, the microbial environment that all this is happening in. And I want to talk a little bit about what we saw from the macro side of things, which is substantially less interesting than microbes, in my opinion. But One of the nice things about the arcs is that they're floating structures, and we know that coral reefs, when they're healthy, are depositing a lot of calcium carbonate, right? They're calcifying. So we can actually weigh the arcs to see if they're getting heavier. And one of the ways we did that was by building this strain gauge that we would use to measure these things as they got heavier. As we can see from this plot over here, this is net buoyancy in kilos, so this is how floaty they are. And you can see that that net buoyancy is going down over time. This is the first arc, this is the second arc. And as it goes down, it really is a strong indicator of calcification. And when we map this to the average calcification rate, this accretion rate is about 690 grams per meter squared per year, which we see pretty frequently in the literature as being pretty common. They're pretty good for a reef. We also had all these new tag-alongs. We had a lot of coral recruitment. They had particularly like weird things like screws, screw heads and stuff, which was really interesting. But we had a ton of coral recruitment on here, and that's also contributing to this increased calcification weight. Coral survival. We moved 8 species of corals equally to all of 4 of these different sites. And of those 8 species, we had about 47% survival on the arcs compared with 24% survival on the control site. So about double. And I should mention here that we are not doing any sort of cleaning or anything like that, like they do in coral restoration. Like we purely deployed all these corals and then let the ecology do its thing, right? And so that's part of the reason why we had lower survival of these corals. And I'll go into, on the next slide, I'll go into a little bit more of why we also think that is. But this is the same data shown on a competing risk analysis that showed that corals were less likely to die or go missing on the arcs than at the control sites. So they survived better, great. They also grew better. You can see pretty clearly from these photos on the left that in a 9-month period, the corals on the arcs looked pretty stoked to be alive, and the corals on the control sites looked pretty not stoked to be alive. And we see that reflected in these growth values here, where we have average living coral tissue with plate. We see a pretty intense spike in the arc's growth, but not nearly as much at the control sites. Now here's where we get, I think, to like potentially some interesting stuff. We know that turf and algae are driving microbialization. So we looked at the percent cover of turf algae on each of these plates. And we can see that on the control sites, it basically skyrockets immediately. And turf algae stays really high throughout the experiment. Whereas at the arcs, turf algae didn't skyrocket as high, and it stayed reasonably low and in control throughout the experiment. So what is going on here, right? We know that microbialization is a dominant challenge at the control sites. Maybe less of a challenge here, but what is going on? What is driving that coral death? And if we look at kind of this picture, these pictures were taken at 18 months. On the right, you see this plate here that's at the control sites, covered in turf and turf consolidated sediment. Not very interesting. These red arrows are denoting corals. And on the left here, you see that this plate is like remarkably complex. It's got all this different shit on it. It's got crustose coralline algae and hydroids, bryozoans, bivalves, macroalgae, cyanobacteria. It's got a ton of stuff, including corals, right? So what this is telling us is that these improved conditions in the midwater are benefiting not just corals, but also all these other organisms, right? Suddenly everybody's having a party. And what's interesting here to me is that right now, one of the main challenges in coral restoration is trying to get over this microbialization hump, right? But one of the questions we have to ask is, okay, if we can fix a lot of these environmental conditions, what's the next challenge that we're going to face, right? Are corals still going to be competitive in a world in which conditions go back to what they were, right? And this might give us a little bit of a vision into that, right? What is going to be the next challenge we're going to face? And it might be benthic competition, and whether or not corals can remain competitive, I think, still remains to be seen. Fish here, you're looking at species richness of fish. You can see that maybe unsurprisingly, because it's a natural reef, the diversity of fish was higher at the control sites throughout the experiment. But the average biomass of fish and the average abundance of fish both skyrocketed as these arcs developed and eventually exceeded that of the control sites by a pretty significant amount, and then stayed that high throughout the experiment. As you might remember from those videos, it's like this crazy fish ball, you know, it's probably because they're attracting these things.
So,
No, go for it.
next slide.
Next
My
slide.
question.
Yeah, so who are the fish? What are they doing? What are they doing, right? We can break them up into what they eat. And so we have piscivores, which eat fish. We have planktivores, which eat plankton. We have secondary consumers which eat anything they can. I think I'm a secondary consumer, and we have primary consumers which are herbivores. And when we look at the biomass of fish at the arcs and the control sites, we can see that pretty quickly these arcs get dominated by piscivores, predators. We know from kind of the beginning of the talk that these predators are stabilizing forces in ecosystems. They're helping lock up all that ecosystem energy into this trophic web. And so the fact that we see a lot of piscivores on these arcs is a strong indicator to us that we've built a system that is, or we've provided the conditions for the system to build itself that is more trophically stable and more canonically healthy. Also interesting, this is when we deployed the arms units, which had all those critters in them. And after we did that, the communities like explode. And so what we think is happening here, we didn't get to do any of the labeling work that we wanted to do, but what we think is happening here is that these arms are providing sources of zooplankton for the base of that fish community, which is then moving up the fish community and providing food. Okay, this work led to a few different papers, and basically the summaries here are that the microbial ecology, biogeochemistry, and the water quality are really creating the environment that selects for these different communities. Midwater arcs were less microbialized than the seafloor control sites, and this improved survival outcomes for corals. After overcoming microbialization, benthic competition seems to emerge as one of our main determinants of coral success. And the arcs were these kind of cool tools that allow us to build these complex reef communities, even in degraded areas. And we ended up kind of running with this and using arcs as these tools in a bunch of different iterations to try and understand how intervening on a reef system drives these different outcomes, right? So arcs are nice because you can change the geometry, you can move them up and down, you can shade them, you can add different biology to them. So you can really change a lot of the ecosystem parameters here. And by doing so, you can learn how that drives ecosystem functions. And I think it's really important to note that in restoration, we have to be integrating science, we have to be integrating experimentation. And so to understand how each of these different variables impacts outcomes to reef communities, I think is really, really important. And arcs seem to be a pretty good tool for this. So we built a new restoration framework for coral reefs based in control theory using this tool. Okay, so that's all I'm going to tell you about the arcs. If you have any more questions, I know I told you a lot, but if you have any more questions, let me know. I'm going to talk just very briefly on a couple of the projects that I'm working here at UH. This one, the UH logo is being bitten by a mosquito because this first one is about mosquitoes. And I'm working in the Amond lab with Matt Medeiros as well on this. And one of our goals here, similar vein of microbes contributing to these ecosystem functions, we're trying to identify the roles of microbes in energy transfer across trophic levels and their impact on food chain efficiency, right? We have these simplified bromeliad systems here, which are composed of, the base of your food web, which is the microbes, and then these mosquito larvae that are the primary consumers, and then, another trophic level and another trophic level as things eat. And the questions we're interested in asking here are what microbes and genes are associated with growth and survival and developmental success in these mosquitoes, which are very cool. Very cool. They're great, and then how the presence of these microbes and genes impacts energy transfer up the food web. So a lot of this work was run before I came by Dr. Nicola Krefall and Iker Iturralde, who's been running the experiments, as well as Fabi Zamora, who's also in our lab. And the first two experiments that we ran, the first one is called 23andMe. It's because we had 23 microbes and one mosquito, or me. And what we did here is we combined the mosquitoes with 12 different microbes. So each mosquito we had these mesocosms that had 12 different microbes and one type of mosquito. And we kind of iterated that 25 different ways to see how you can mix up the community and how that drives changes in mosquito survival and growth. And so the goal here is to basically identify the microbes with positive and negative associations on mosquito growth and survival. From the results of that, we pulled out 4 microbes that we call K, P, C, and N. Homomonas, Posiconia, Pseudomonas, Homomonas, Nova Syngylium. And then we actually ran fully factorial placket vermin designs on these mosquitoes again. So instead of running it a bunch of different ways, we had all 4 microbes We had the triplets, the doublets, and the singlets. The goal here was to kind of dig into specifically a little bit more of the genes, the positive and negative associated genes with growth, survival, and developmental success. And our final experiment, which we just ran recently, maybe final, maybe not, maybe it could go on forever, is that we call Toxo, which is about the, we added in a second trophic level mosquito called Toxobronchites, which eats the Culex. And so now we have an even more complex system because we love complexity. And we had basically the same design, a little bit reduced, measured toxo at the biomass, or toxo biomass at the larval stage. And the goal is to identify now gene positive and negative genes associated with growth, survival, developmental success, and food chain efficiency. So that's what we're working on right now. It's taken us a lot of time, which is why I'm not going to show you any data today. But I'm sure there will be time for that. And then lastly, I'll just touch briefly on one of the projects that I'm excited about starting up here with a couple of people who are actually in this room, Aaron and Maggie. And this is back to the ocean. And the idea is that open ocean productivity, there's this really rich community in the open ocean phytoplankton that's producing like 50% of all the oxygen on Earth. It's crazy. And the phytoplankton then get eaten by zooplankton, which get eaten by bigger and bigger things. So there's a lot of biomass out there. There's a lot of production. And all of that production has to go somewhere, right? So it's either remineralized to the atmosphere or, respired to the atmosphere, or it's exported and it starts sinking in these aggregated particles called marine snow. And this is called a biological carbon pump, which is storing a lot of this material in the deep ocean. And along the way, microbes, fungi, bacteria, everything are really determining how much of it is exported or respired back to CO2. And we particularly know like nothing about fungi and what they're doing here. So this is one of our main questions. We do know that on land in terrestrial systems, fungi are responsible for taking these big, robust, hard to degrade things like cellulose and converting them to smaller, easier to eat things that bacteria then eat, right? So our goal here is to kind of disentangle these bacterial and fungal roles in the degradation of marine snow using something called stable isotope probing and metatranscriptomics. Main questions are who the primary fungal and bacterial degraders are of these polymers. Do bacteria and fungi compete or cooperate for these substrates? And how much of total flux can we attribute to these different compartments? We're doing this here. We have a really great long-term study site here in Hawaii called Station Aloha, which is just north of Oahu. Goal will be to collect marine snow particles on the HOTS cruises, and then basically, run these community labeling studies where we feed them these isotopically labeled carbon-13 substrates, which if they eat them, then get incorporated into their DNA. And then we can extract that DNA. We can separate the heavy and the light fractions, the ones with isotopes in it, using density gradient centrifugation. And then we can sequence it to see who's there and who's been munching. And so that's the goal. That's what we're doing. We have some cool ideas for that, but we are still, in the very, very early stages of conceptualization. So that is, I think, enough for me to tell you guys today. So I want to thank everybody in my lab and my mentors, new and old, and as well as all my funding sources. So yeah. We need to